या फिर फिर से वही बैक स्ट्रेट करके बैठो बेंच में। मैं बोल नहीं रही हूँ, आई एम सेइंग वही जो now you are in the 10th, so you have attended this type of classes 8th में ना 7th में 8th और 9th तो virtually गया ना almost ना। इसी तरह से बैठना 2-3 घंटे एंड स्कूल में 6-7 आवर्स। अच्छा लग रहा है कि नहीं अच्छा लग रहा है? अच्छा लग रहा है? Very good. So which schools are here? साधु वासवानी तो होगा ही। How many of you from साधु वासवानी here? साधु वासवानी सब बदमाश लोग हो तुम लोग। और दूसरे स्कूल्स कौन से हैं? खुदा नेहरू का वहाँ से तुम यहाँ पे क्यों आए? अच्छा ओके। और 7th डे छात्र नहीं जाना पड़ता तुमको स्कूल नहीं? what the meaning of that term 7th day meaning पता है? 7th day हाँ उसको 7th day क्यों बोला जाता है पता है? जिसके साथ से स्कूल में पढ़ रहे हो उसका मतलब ही नहीं पता। क्वेश्चन स्टार्ट हुआ है स्कूल में। Which are the chapters that has been started? Cell cycle completed, genes started. Cell cycle completed and genes is started. Genetics start हुआ है। Now for polar it is absorption and trans completed. I know. They always start in the middle of the chapter. तुम दोनों पोलर के हो? 7 day. अच्छा पोलर 7 day. 7 पोलर. And for how many of you portion already started? 7th day polar 7 day. अच्छा तो basically 7th day पोलर तुम लोग का? पोलर. साधु वासवानी में भी they have started for this course. So they have not talked about any exam right now, no? Yes. Any exam? Yes. For polar and 7th day any exams? Yes. कब है? May 4. And today we are April 4th. अभी 1 महीने का टाइम। Why I am asking you this is, should we go sequentially? Yeah, because generally for 9th and 10th, as per the requirements of the school, we generally take up all the chapters we take. So now, 2 schools that have started with absorption, and the rest you all have started with cell cycle. Cell cycle खत्म हो गया, अभी बाकी सब ना. So, and for you अभी कोई exam है? तुम लोग का भी होता है तुम लोग का भी एग्जाम दिए हैं। So since your exams are not recently within a period of month कोई एग्जाम नहीं है, so in that case I will prefer to start from the first chapter and cell division cell cycle will try to do as much possible. After that I will skip to these chapters. Genetics में बाद में करूँगी। Genetics क्योंकि they have started now. Variation plants computers. So genetics हम बाद में करेंगे। Let's see when other schools starting the genetics एक difficult concept है वो भी एक बार 2 बार लेना पड़ेगा। So पहले ये 2 चैप्टर करेंगे। अच्छा now in my absence I think sir has taken 1-2 lecture sir ने लिया हुआ है। तो कौन सा चैप्टर सर ने स्टार्ट किया? Cell division. Cell division cell cycle, right? नहीं cell नहीं, cell is just for introduction. उसमें बोर्ड में कोई क्वेश्चन नहीं आएगा। For the first chapter, cell. This is just a revision to make you acquainted with the concept of cell. क्योंकि next chapter cell division है। ये chapter cell study है, ठीक है? So first chapter we are not doing. So now being the first class for me, already known, I'm going to teach the biology portion here. And next is one request from my side after I welcome all of you to this academic session. I am very much sure that you all will have a smooth and a very productive period here. 10 standard. Are you tensed? कोई टेंशन नहीं है. You should enjoy the process of study. You should love to study. अच्छा लगता है पढ़ाई? मम्मी मारती है तभी जाके पढ़ने बैठते हो. अच्छा लगता है पढ़ाई? सच है. You should love studies 10th, 12th, no big boulder on your head. If you love the process, interact more with teachers, interact with your friends in matters of studies, things will be very simple. पढ़ाई तो पढ़ना है. ये मत बोलना घर जाके मैम ने बोला है कि कल इंटरैक्ट करो तो सब थिंग्स will be simple. पढ़ना तो है ही. इसके साथ the more you interact, ऐसे नहीं कि मैं आके पढ़ाती हूँ चली जाओ पढ़ाओ पढ़ाते अगर you ask no questions कोई interaction नहीं. Try that in the class you learn maximum. जो भी डाउट है एग्जाम के पहले क्यों पूछना है, फिर पूछो every day. So that's the system that we always follow here. हर दिन I will teach you something, I will give you homeworks. अगले दिन जो भी बोलेगा कि मेरा स्कूल में ये था, that's the reason I could not do. Unless and until you have any medical reason, I don't want any reason. स्कूल में तो सारा साल एग्जाम और टेस्ट और असेसमेंट एक्सेट्रा रहेगा. That does not mean that you will not do the work here. यहाँ पे जो भी हम होमवर्क रहेंगे, any subject, please find out time to do it. Right from now, you should know how to plan your time. आज सुबह बैठे एक चैप्टर लेके पढ़ने के लिए बार-बार वो पेज खोल रहे हो, सेम चीज पढ़ रहे हो. Half of the day gone, you have not learned anything. That should not be the way for a 10th, 12th standard student. Plan your day, plan your time, so you'll be able to do everything in the 24 hours. Okay? So, we will try to make everything very simple, your school friendly, so that every time you are well prepared for your school exams, your tests, and for that you have to intimate us well ahead. These are the exam dates, these are the portions, so that in the class we can take a call and take your portions ahead. Okay? ऐसा नहीं कि अभी एग्जाम के 2 दिन पहले तुम आके बोलो कि these are the 6 chapters that are finished, so that will be difficult for most class. So well ahead of time, if not me, the coordinator, or directly us in the classroom, intimate करो when are your exam dates. Because see, it's a mixed bag. Different schools के different requirements रहेंगे. But that's not a problem for us. We have been always doing this. Off and on, we'll try to satisfy every school. Okay? So that's nothing to worry. As we start, hope we go very smooth. All of you have time? All of you have time? Make sure that you sign in this sheet also every lecture. Start with the concept of cell and cell division. As I said, so first of all we'll take up these 2 chapters, cell division, cell cycle, and then I'll move to your portions, do 2 chapters. This way how we have to do so that all of you are satisfied with the portion, prepared for your school exam. The first concept that we start with, and one more thing is, I think all of you use Selena in your school. Shrijan, which school is using Shrijan? Yes, no, you have the Selena. Sadhu Basrani also nowadays use Selena. Which school is using Shreya in your school? No. All of you here have Selena. Evergreen. Evergreen. अच्छा. इसके portions देखो. Evergreen and this I think more or less same. This is Selena more or less same. Earlier, Southwestern they used to use Shreya, but now I think Avalon Heights. No, Avalon Heights is not here. So now we start with this concept of cell, chromosome, chromatin, DNA. That's the first part that we're going to start with. Turn to your chapter, turn to your textbook. The second chapter we are going to start with, that is structure of chromosome, as the first part of the chapter. As we start with this concept of structure of chromosome, first of all, we have to find a link between certain words like chromosome, chromatin, chromatid, nucleus, DNA, etc. So I start with these few words as an introduction. Nucleus, let us start with this, nucleus. And the previous standard already talked about the word cell, no? What are the two prominent components, or if I ask you, what are the two prominent compartments of a cell? What is the answer? Is there one? What are the two prominent compartments of a cell?
Nucleus and plasma.
Cytoplasm. Nucleus and cytoplasm. So that means I am talking about this nucleus, and if I try to understand that, say suppose this is a cell, animal cell, and this animal cell has got a well-defined nucleus, and inside the nucleus there is the hereditary material. So this part is what the nucleus is. Nuclear membrane. Entire chamber or the compartment can be termed as a nucleus. Any type of a cell which has well organized nucleus. कौन सा type of cell है? Which type of cell it is? Eukaryote. A type of a cell which has got organized nucleus. Eukaryote. Okay. So I am talking about this concept of a cell, जो previous year में तुमने पढ़ा है about the concept of cell in the Untitled Chapter. हर एक cell में there are two major compartments. इसको बोलेंगे cytoplasm and इसको बोलेंगे nucleus. Always to understand the two major components of a cell, cytoplasm, nucleus. Inside the nucleus, hereditary material is present. वैसे तो inside the nucleus, what do you call? Do you remember the name of this matrix जो inside the nucleus है? What is this matrix called? Very good. Nucleoplasm. Nucleoplasm has got the thread-like structures. अब ये thread-like structures जो है, these can be given different terms like chromatin at times, chromosome at times, and even chromatid. So ये तीनों almost same ही structure के different names at different stages. Something like our growth stages. We have got the infant, the infancy, and then the adolescence, and same ही structure है, but at different stages of the cell. इसको अलग नाम दिया गया है. Okay, we'll try to understand. Try to remember the connection between all these words. So I am basically talking about the nucleus right now. Nucleus के अंदर hereditary material. Now when this cell is a young cell, what is the meaning of the word young cell? When it is just formed after the process of division. When it is formed after the process of division, something like I am talking about a cell nucleus here, and the hereditary material is like this. This cell will divide. एक से दो बनेगा. The cell will divide. When this cell will divide, this is a young cell. This is a young cell, and this young cell will undergo the process of growth and maturation. In the concept of biology, we will always use the term a mature cell and a young cell. तो ये समझ में आना चाहिए कि what is the meaning of the word young cell किसको बोलेंगे? A cell which is just newly formed after the process of cell division. And the cell which is maturing and getting ready for the process of division again, and is getting ready for the process of division again, इसको हम बोलेंगे mature cell, and this is a young cell. तो वैसे तो young cell और mature cell में size में तो difference थोड़ा बहुत होता ही है, but apart from that, the major difference we come to understand in terms of its hereditary material. In terms of its hereditary material. समझो, if I say ये जो hereditary material जो अंदर मैंने draw किया, maybe now I can draw that the hereditary material is something like this. Can you count कितने thread-like structures है? You cannot. मेरा diagram में defect नहीं है. Actually you cannot. When we are talking about any young cell, when we see under high magnification of the microscope also, we cannot understand how many thread-like structures are there in the nuclear regions. But अगर mature stage में देखा जाए, कोई भी cell, you can easily count the thread-like structures. You can easily count the thread-like structures. Now this is mature, ready to divide, ready for division. Apart from that, is there any other difference between the young and the mature cell? No, cell organelles and cell membrane लगभग same है. Composition will different. So now this type of hereditary material जो मैंने draw किया यहाँ पे extensively long and fine, इसको बोलेंगे chromatin or chromatin threads. Chromatin या तो chromatin threads. अब जब ये completely थिक हो गया है and visible हो गया है, prominent हो गया है, इसको हम बोलेंगे chromosomes. तो definition क्या है? What are chromatin threads? Extremely long and coiled, thin thread-like structures that contain genetic information. But वही thin thread-like structure जो इस तरह के रहते हैं, now they become condensed. इस तरह से condensed हो जाता है, and that is the reason हमें visible होने लगता है. They start coiling. That's the reason they appear broader and thicker, shorter also. They appear thicker, they become shorter. तो पहले तो लंबा सा thread था, extensively coiled था, so that's the reason we were not able to count how many of them are there. But now that they have undergone the process of extensive coiling and condensation and shortening up, and they appear broader, now we are able to visibly distinguish them. Chromosomes. So what is the basic difference? Chromatin threads, long thread-like structures, extensively coiled, extensively thin, which contain hereditary information. Chromatin threads which are extensively condensed, coiled, appear shorter and broader, are called chromosomes. And are they present in the same cell? Is it possible that they both are present in the same cell? Chromatin and chromosome कभी भी एक ही सेल में present होगा? जोर से बोलो ना. मास्क खोलने की जरूरत नहीं है. Voice loud. No, not possible. I've been repeatedly telling always to remember chromatin threads are present in young cells. Chromosomes are present in mature cells. ठीक है? Clear है? Now, if I say that ये जो chromosome है, for simplicity, we generally draw a chromosome this way. जहाँ पे this is a condensed region, है ना? इसको आर्म्स भी बोला जाता है, यहाँ या तो क्रोमैटिड भी बोला जाता है. आर्म्स या तो chromatid. So what is a chromatid? Arms of the chromosome which are extensively coiled and thick. ठीक है? So तीन terms all related, but इसमें chemical composition की कोई difference नहीं है. इसमें is there any difference in the chemical composition? Nothing. No difference in the chemical composition. See that the same thing I'm trying to show you also here in this picture. We have been talking about the nuclear region which has got all these hereditary structures. See here. Now if this is the diagram that I am showing you of a cell, can you tell me? Am I showing you the diagram of a young cell or a mature cell?
Mature cell.
It's a mature cell because you can easily count the chromosomes, how many of them are there. But say suppose if I am showing you, let me see if I have any other. If I am showing you something this way, this is comparatively a less mature cell because the threads appear thinner and coiled. So this is what we are going to come across in the next concept also when we study another concept of cell division. How to identify which cell it is, which stage it is. By the coiling of the chromosome we can identify. Okay. So this is what I said, that these three terms, are they related to each other? Yes. Under what concept? Maturity of the cell. And next is what? Composition. Is there any difference? No. What are they made up of? DNA. Whether I talk about chromosome or I talk about chromatin or I talk about chromatid, composition is the same. What is it? It's a macromolecule. It's a macromolecule. And what is the full form? पता है? अरे ये तो जानते हैं. DNA. What does this DNA stand for? Deoxyribonucleic acid. Deoxyribonucleic acid. याद रखना है, the full form of DNA, deoxyribonucleic acid. Okay. Now, what is this? Is it acidic or basic in nature? Acidic. Since it is acidic, it has now this deoxyribonucleic acid that I am talking about, I already said. It's a macromolecule, meaning it's a complex molecule, big molecule. And this big molecule, this macromolecule, being acidic, it will always have a tendency of binding with a basic molecule. Acidic components will always have a affinity for basic molecules. Is not so? Negatively charged will always have a tendency for positively charged molecules. Exactly the same thing is happening. This deoxyribonucleic acid has a tendency for binding and winding Around some basic proteins. And what are those proteins? These proteins are called histones. That is the reason for this coiling. We'll talk about the detailed structure later. But try understanding step by step क्या हो रहा है, what are the connections of one term with the other. Deoxyribonucleic acid and histone, एक acidic है and this is basic. This is a protein. Histone is a protein, and in the 9th standard you had studied nutrition. A chapter was there, no? Nutrition. There you had studied about proteins. Proteins are made up of
Carbon, oxygen, nitrogen and hydrogen.
किसने बोला? अच्छा. Carbon, hydrogen, oxygen, nitrogen. Repeating units को क्या बोलेंगे? प्रोटीन के repeating units को क्या बोलेंगे? कौन बोलेगा? प्रोटीन के repeating units को क्या बोलेंगे? Let's start with nutrition में था. Amino acids? हाँ. प्रोटीन के जो repeating units है, if I talk about a long protein chain, ऐसा होता है ना? There one single unit amino acid. तो amino acid तो positively charged भी होता है, negatively charged भी होता है. So when I am talking about histones, I am talking about protein which has all positively charged amino acids. So ये DNA जो है, this starts coiling or winding around this histone proteins, and that is the reason for the coil. Why should this chromatin threads coil? Because the DNA starts getting bonded. I mean, it has a basic affinity. Starts winding around this basic proteins. Now, when we talk about this DNA molecule, first of all, हम DNA के बारे में सीखेंगे. Then we'll go with this complex structure. Now, when we talk about this DNA molecule, how does this DNA molecule look like? However, now that I said कि ये जो structures है, or if I say now something like this, ये जो structure है, ये कोई भी एक छोटा सा region, if we try to magnify and see, how would it appear? As I said already, chromatin हो या chromosome हो, basic composition is DNA. कैसे दिखता है DNA molecule? DNA molecule is a helical structure. DNA molecule is a helical structure, and this particular helical structure has basic properties. We are going to learn these properties now. पहले तो both these thread-like structure, they are called what? Strands. Strands. DNA molecule is double-stranded. What is the meaning? Made up of two threads. Made up of two threads. So, what are the terms that we talk about? First of all, I said these are the strands. Let me straight away talk about the terms that you should know. The first is we say DNA molecule is double-stranded. Meaning? Made up of two threads. DNA molecule is helical. Meaning it is not straight linear. Curved. It is curved. Helical. DNA molecule is anti-parallel. Anti-parallel. देखो DNA molecule helical है. See the DNA structure that you have in your book. Here we call it draw करोगे. It comes in the paper for drawing. Get acquainted with the structure, and as I am explaining, also try to understand. Now, this DNA molecule, एक और property ये है anti-parallel. अब anti-parallel मतलब, if I see how does this strand gets formed, it is something like this. That if one strand is growing in this direction, the other strand will grow in this direction. Meaning, This is a macromolecule which is exactly like protein made up of some repeating units. जैसे मैंने अभी बोला है कि एमिनो एसिड्स लिंक होके एक बड़ा प्रोटीन चेन बनता है. सेम लाइक दैट डीएनए चेन जब बनता है इट इज समथिंग लाइक बाय द एसोसिएशन ऑफ सर्टेन रिपीटिंग यूनिट्स. So DNA chain भी बनने के लिए some repeating units has to come together get bonded. तो ये जो coming together and getting bonded अगर एक का इस डायरेक्शन में हो रहा है तो दूसरे का इस डायरेक्शन में होगा. That's the reason. It's something like on a parallel road, two vehicles are moving. One is going this direction, the other is coming this direction. Or talk about the tracks of the train. Something like एक इस तरफ जा रहा है, दूसरा इस तरफ जा रहा है, but they are connected. Same like that. These two strands remain connected, remain connected, remain connected, remain connected like this, almost looking like a ladder. Almost giving the appearance of a ladder. So this is how it generally appears. And what are these bonds basically? The bonds which are there between them are basically hydrogen bonds. Now the question is कि ये जो ladder like structure जो बना है, this ladder like structure is made up of certain repeating units. Let me start from here. I say that एक repeating unit यहाँ है तो दूसरा repeating unit यहाँ होगा and there is a connecting bond and this bond is known as the hydrogen bond. तो ये जो मैंने green globular structure draw किया है, I am trying to show you that this is a repeating unit. तो इस तरह के repeating units काफी सारे होंगे. Let me draw somewhere one more here. Let me draw one more here. Let me draw one more here. And this is all throughout. So दो strings है, दो strands है, दो thread-like structures है. दोनों में certain repeating units has to be there and the two repeating units remain connected. And that generally gives a ladder-like appearance. तो ये repeating units को हम क्या बोलेंगे? What is this repeating unit known as? Very good. Nucleotides. ये repeating unit को क्या बोलेंगे? Nucleotide. This is a nucleotide. ठीक है? This is a nucleotide. So now, when we talk about the structure of the DNA molecule, so this is what you get to see. अगर DNA molecule को straight कर सकते हैं, so that will always look like this, a ladder, है ना? Where these are basically the two strands, and यहाँ पे कुछ repeating units है which remain connected by bonds, and ये hydrogen bonds वो stairs जैसे एक appearance देगा. So now next is what we try understanding the next concept. अच्छा, यहाँ पे still something more to understand. I said ये जो repeating units है, these are called nucleotides. Now, what are these nucleotides made up of?
Phosphates, nitrogen, base, and pentose.
So अभी whenever we are talking about a nucleotide, what is the definition? First of all, repeating units of the DNA molecule. What is a nucleotide? Repeating units of the DNA molecule. Now, to understand this concept, let us see here. The nucleotide. What are the components? I said, let us start with the components first. The components of a nucleotide. The components of a nucleotide. The first component is pentose sugar. The first component is a pentose sugar. What is the meaning? 5-carbon sugar. This pentose sugar is known as what? Deoxyribose. pentose sugar. Okay? Now this pentose sugar will be linked with a component, and this component will be considered, or this component will be known as nitrogenous bases. Nitrogenous bases. I'll show you something here. Now that much structure you don't have so much in detail, but little bit you should know about this. Let me see if I have this. I don't have this here. Now, when we are talking about this nitrogenous bases, first of all, simple to understand what is it. These are also chemical components. And this nitrogenous bases may come in two categories. Okay. This nitrogenous bases may come in two categories. What are the two categories? Let us see. The two categories are one type of nitrogenous bases. Would generally have a structure something like this. Would generally have a structure, a single ring with nitrogen in the first and the third position, and the rest are carbon, hydrogen, oxygen compounds these are. So when we are talking about this, so when we look into one type of nitrogenous base which has got a single ring structure, we talk about this as pyrimidines. There are another type which has a double ring structure, and these are known as purines. Two types of nitrogenous bases, pyrimidines, purines. So which are the purines? Purines are adenine and guanine. Which are the pyrimidines? Cytosine, thymine. Now, when we are talking about cytosine and thymine, can you see in this figure there are some letters written? Can you see here A, T, G? Some letters there. Can you see there? So they are talking about the nucleotide with a specific nitrogenous base. So let us complete this. So this nitrogenous base can either fall to this category or may fall to this category. Purines, double chain, double ring, and pyrimidines, single ring. अब कौन सा component बाकी रह गया? The phosphate. The phosphate. So when we are talking about phosphate, ये बाकी रह गया. So अब ये तीनों component जब एक साथ है, this is called a nucleotide. So what are the three components of a nucleotide? Deoxyribose or a pentose sugar, nitrogenous base, pentose phosphate group. अगर phosphate group नहीं रहेगा, without the phosphate group, अगर phosphate group नहीं है, then what it is? It becomes a nucleoside. With phosphate, it is a nucleotide. आया समझ में? When the nitrogenous base and deoxyribose only combine, they form nucleoside. So how many types of nucleosides are possible? How many types of nucleosides are possible in the DNA? 4. कैसे 4? Sometimes nitrogenous base will be cytosine. Sometimes in one nucleotide. In one nucleotide, I will come to that structure again. In one nucleotide, I am talking about a single nucleotide only. In one nucleotide, sometimes nitrogenous base, yes, nitrogenous base will be cytosine, sometimes adenine, sometimes thymine, sometimes guanine. एक न्यूक्लियोटाइड में दो नाइट्रोजनस बेस कभी होगा नहीं, ओनली वन. दैट मीन्स अगर मैंने ये सब को न्यूक्लियोटाइड बोला है, दैट मीन्स समटाइम्स इन वन कभी एडेनिन होगा, दूसरे में थायमिन होगा, एक में ग्वानिन होगा तो दूसरे में साइटोसिन होगा. पर क्या मैं ऐसे लिख सकती हूँ थायमिन एंड ग्वानिन ऐसे लिख सकती हूँ? नो, दैट इज डेफिनेटली रॉन्ग, क्योंकि एक प्यूरिन विल ऑलवेज पेयर विद अ pyrimidine. एक प्यूरिन और pyrimidine का ही हमेशा पेयरिंग होता है. So adenine basically pairs with thymine and vice versa. Guanine basically pairs with cytosine and vice versa. That is standard. But कभी ऐसा होता है कि guanine has paired up with thymine, होता है? That's an error. Error. इसको error माना जाता है. That's not a normal condition. जबकि if certain things are there in the DNA, the protein produces will be a little bit different from what is normal. See, that's the reason we all have some deficiencies of some proteins or excess of some pigments or etc. No? So that way we don't become abnormal, but then हमारे सबकी बॉडी में कुछ ना कुछ deficiency, कुछ ना कुछ disorder है, छोटा बड़ा. That's not a major ailment that we are talking about, but ये इस तरह के errors हैं हमारे DNA में. इसको क्या बोलते हैं? बेस पेयरिंग. Nitrogenous base pairing. So what is the base pair rule? Adenine always pair with thymine, guanine always pair with cytosine. इसीलिए तुमने ये वाले देखो A and T always, G and C always. देखा? A and T, G and C. Clear है ना? So what did we learn? That when these three components come together, the nitrogenous base is why are you putting words in my mouth? Nitrogenous bases, pentose sugar, phosphate group. जब ये तीनों components एक साथ आ जाएगा, उसको हम बोलेंगे nucleotide. और nucleoside को बोलेंगे phosphate. जब नहीं attach होगा, then nucleoside. ठीक है? Then how many types of nucleoside? 4. How many types of nucleotide? 4. Now in a nucleotide or in all the nucleotides of the DNA molecule, if it is time tell me, I'm not telling you. In all the nucleotides of the DNA molecule, which is a variable component? In all the nucleotides, which is the variable component? ये हो गया first component, ये हो गया second component, ये हो गया third component. If I am talking about there are 4 nucleotides, one with adenine, the other with thymine, the other with cytosine and the other with guanine. तो ये चारों जो nucleotides है उनमें standard component है deoxyribose. ये तो हर एक में same है. Phosphate group हर एक में same है. तो variable component कौन सा है in a nucleotide? Nitrogenous bases, क्योंकि वो ही कभी adenine होगा, कभी thymine होगा, कभी cytosine होगा, कभी guanine होगा. Phosphate और deoxyribose शुगर तो एक ही है सब में. आया समझ में? So now, what are the basic concepts to understand? You have a basic question. What are the repeating units of the DNA molecule? What are the components of the nucleotide? Why is the DNA molecule known as a macromolecule? That's a very simple question you get in a paper. Why is it known as a macromolecule? Because it is made up of various components. 1, 2, 3, and the nitrogenous bases itself can be of 2 types. If they are double ring purines, for example adenine guanine, and if they are single ring pyrimidines, for example cytosine thymine. So it is not a simple basic carbon hydrogen nitrogen oxygen का structure. It's a complex molecule, hence it's a macromolecule. And some of the properties there, you remember Y helical, Y antiparallel. And what generally forms the ladder of the staircase of the DNA molecule? The pairing up of the nitrogenous bases. The pairing up of the nitrogenous bases. देखो, one nitrogenous base, actually when we draw the structure, you will always come across the one nitrogenous base generally gets linked with the other. For example, here, if I show you here that this is one nucleotide, what will be the structure? Yes, this nitrogenous base, if I show you that this is with adenine and this will be the other nitrogenous base thymine, and the rest of it will come here, something like, if I talk about the pentose sugar will be not this way and the phosphate group will be not this way. But why did I draw it the other way? Because it is anti-parallel. If one strand is moving this direction, the other strand will move in this direction. The orientation of the strands will be opposite. One is this way going, the other this way coming. So that is what we understand as the concepts of the DNA molecule. Okay, the DNA molecule. Take out your notebook and very fast draw the double helical structure of DNA. Draw the double helical structure of DNA. That will generally be getting a paper for 1 or 2 marks. Neat diagram, very simple diagram it is. Not with a pen, with a pencil. No shading coloring needed. Only 2 strands and the ladder-like structure and the ATGC shown. That's it. Write the chapter name, write down the name of the chapter. And the rest of the information, major group, minor group, that's not given in your portion, so I am not teaching also, so no need of that. Pencil. Bring your pencil for the next few classes, क्योंकि सेल डिवीजन में भी काफी सारे डायग्राम्स ड्रॉ करना है. See, drawing the diagram of the DNA is very simple. कैसे draw करोगे? ऐसे लेना है, ऐसे लेना है, ऐसे लेना है, ऐसे आना है. And parallel draw करो इसके साथ. ऐसे आओगे, simple. एक stroke में diagram हो जाएगा. उसी के साथ साथ parallel आना है. Half of the page में diagram complete होना चाहिए. Any diagram that comes in the paper, half of the page में complete होना चाहिए, but neat होना चाहिए. Pencil drawing होना चाहिए. अरे ये तो आर्टवर्क हो गया रे बाबा. इतना पतला नहीं ड्रॉ करना है. It should be reasonably thick also. Diagram should be ऐसे नहीं है तुम्हारे पास. हाँ ऐसे नहीं है. This much thick it has to be. Parallel. 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 Alright? No, actually ऐसा नहीं draw करना. Don't draw this, huh? I'm showing it. ऐसा नहीं draw करना. ऐसे नहीं ड्रॉ करना. हाँ देखो मैंने उसको ड्रॉ करके दिया. इस तरह से ड्रॉ करना है. हाँ कैंडिडेट यूज भी करा मैंने प्रॉब्लम. ऐसे करो, ऐसे आओ, ऐसे. इस तरह का वाइट W एक ड्रॉ करना है. ब्लैंक एक W ड्रॉ करना है. एंड देन यहाँ से फिर से ले लोगे, फिर ऐसे आ जाओगे, फिर ऐसे आ जाओगे, फिर ऐसे आ जाओगे, फिर ऐसे आ जाओगे. हुआ कि नहीं? हाँ, sharp नहीं है, blunt draw करना है. थोड़ा blunt होता है. The edges are never sharp. सिंगल लाइन ड्रॉ करो, डबल लाइन अगर डिफिकल्ट लग रहा है तो डबल लाइन मत करो, सिंगल लाइन ही इनफ है. एंड ये जब नाउ फॉर अ 10th स्टैंडर्ड स्टूडेंट चैप्टर नेम लिखोगे. Your book has all the important contents, but whenever you are listening, sitting for a lecture, important points यहाँ पे लिखोगे. So that will also give you an idea कि what are the topics covered and how much, how you should study. Yes. हो गया? This way, this way, this way. तो इस प्रकार का लगभग ऐसे आना चाहिए. Next, this way, this way, this way, this way. Now if you want to draw double, जैसे यहाँ पे इन लोगों ने double standard draw किया, 2 line diagram इसको बोलते हैं. तो अगर तुमको दो करना है तो इसको तुम थोड़ा शेप करके मोटा कर दो, या तो तुम सिंगल ऐसे करोगे then also it's okay. अरे ये ऐसे कैसे, ये कहाँ से ड्रॉ किया तुमने? अगर तुम सिंगल लाइन करना चाहते हो तो इसको थोड़ा सा डिटेल से ड्रॉ कर लो. डबल लाइन करना चाहते हो तो इसके ऊपर से एक और पतला सा पैरेलल डबल लाइन करना पड़ेगा. देन प्लीज गेट दिस टाइप ऑफ रिबन लाइन. स्ट्रेट लाइन करोगे तो भी ओके है, रिबन लाइन करोगे तो भी ओके है. हाँ ये ओके है, नॉट बैड. बट आपको क्वेश्चन व्हाई लाइक दैट, क्यों ऐसा? हाँ लोअर स्टैंडर्ड्स में कभी भी एप्पल मैंगो ड्रॉ नहीं किया था? नहीं, एप्पल मैंगो टीचर ने कभी ड्रॉ नहीं करने दिया, SND ने कभी ड्रॉ नहीं किया. ऐसा घर पे ड्राइंग क्यों है तुम्हारा? पेंसिल भी शार्प नहीं है. YW फिर उसी के पैरेलल. अब तुम ऐसा करो तो भी ठीक है, रिपीटेडली आप कह रही हो. या तो तुम अगर ऐसे रिबन जैसे करना चाहते हो तो इसके साथ साथ ऐसे करके करोगे, थोड़ा शेप करोगे, रिबन बन जाएगा. तो ऐसे ट्रैक भी करोगे तो भी ना करें. चार पांच पांच DNA ड्रॉ कर रहा है वो, और एक भी सही नहीं है. हाँ अभी तुमको ड्रॉ करके दिया था ना मैंने. हाँ आर्ट में तुमको क्या ग्रेड मिलता था? ए मिलता था, और ये हटक है तुम्हारा. उधर का ओपन हाउस था, तुम लोगों का ओपन हाउस था नहीं? अभी ऐसे बॉन्ड ऐसे दे दो इस तरह के, and then as it is given in your book, that way also you can draw. In your book they have shown this way. जैसे यहाँ पे A लिखा तो यहाँ पे T लिखा, इस तरह से it is shown in your book. ऐसे भी कर सकते हो. Such a waste of resource. हाँ? पेरेंट्स ने नोटबुक खरीद के दिया तो इसको वेस्ट कर रहे हो. प्रैक्टिस कर लो. इसमें डिफिकल्ट क्या है? एक वाइड W ड्रॉ करने में डिफिकल्ट क्या है? ऐसे 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 एक वाइड W से ड्रॉ करना है, एंड उसके पैरेलल एक और ड्रॉ करना है, उसके पैरेलल एक और ड्रॉ करना है. Done, all of you done? ए छोटा सा टी लिखो, बीच में बॉन्ड लिखो. तो it is something like इस तरह के पूरा लेटर्स नहीं ड्रॉ करो. Something like A C इस तरह से कर लो. So it looks better. हाँ चलेगा, वो भी चलेगा. We have written down this one A B D C वो लिखा है ना. ठीक है. Make it bit clear. ठीक है. वो बीच में you may just देखा तो बैठो. You may अंदर खाली लिख लो या तो बाहर अगर तुमने लिखा होती है या तो अंदर लिख लो और अंदर तो that also ठीक है. और डायग्राम थोड़ा सा और बड़ा थोड़ा सा फ्लैट ड्रॉ करोगे ना then you get enough space to write this few letters for example. What? ऐसे नहीं करो. ऐसे अगर क्योंकि when you are drawing a diagram for your exam तो ऐसे करोगे ये जो तुमको हाइड्रोजन स्पेस हाइड्रोजन बॉन्ड्स एंड न्यूक्लियोटाइड्स दिखाना है straight away you can do this like this. This is easier for you. यहाँ पे 2 यहाँ पे 3 हो गया. हाँ यहाँ तक ठीक है. ये पोर्शन रख दो. It's 35 तक है ना. It's 35 है ना. हाँ it's 35. Done. We are not starting a new topic now, but all of you should finish with this work. Done. Inside, all of you should at least mark. Inside the nucleotide pairs ATGC, at least few should be marked. Few should be marked. Next class, all of you should have pencils with you, erasers. We are going to study the concept of cell division and उसमें diagram रहेगा. Cell division के stages draw करने हैं. So you should have pencil, erasers. ठीक है? We will wind up here. Continue again with the concept of cell division next class. Chromosomes little bit more left and followed by cell division.